Welcome to the Niche Podcast, your weekly 5-minute rundown of the biotech, clinical research, and applied science industries. I'm your host, Dr. Noah Goodson. Today on the Niche, we'll be discussing 2 deaths in a gene therapy trial, new COVID-19 vaccine guidelines from the FDA, and recent developments in CRISPR-Cas9 gene editing technologies. The Niche is brought to you today by the Scope Method, LLC. The Scope Method helps companies develop clear vision and strategic processes. Whether you need fresh eyes on your data, independent risk assessment, or are pivoting into a new therapeutic space, the Scope Method will help you focus close to re-examine what you know and look ahead to where you want to go. Find out more at thescopemethod.com. Drs. James Wilson and Terry Flott put out a brief report last week after 2 children enrolled in a phase 1 trial for X-linked myotubular myopathy passed away. X-linked myotubular myopathy typically causes individuals to experience a low quality of life, and childhood mortality is the most common outcome of this debilitating genetic disorder. A novel gene therapy had proven safe and effective in mice and non-human primates. However, the 3 patients given the highest dose of the AAV8 delivered gene therapy developed a severe hepatotoxic response, though the details are not well understood at this time. This immune-mediated response from the liver resulted in 2 of the individuals passing away. Notably, patients with the normal AAV dose did not experience these serious adverse events. The trial is currently stopped. Whether or not they'll be able to proceed after this will likely depend partially on the success of the normal dose patients, as well as identification of the cause of death. The delivery of pricey, rare disease gene therapies has continued to fuel the flow of capital from top pharma players as well as VC-backed ventures into the field. The FDA's approval of the $2.125 million per dose gene therapy Zolgensma last year has kept hope around these mega cost treatments alive. Despite setbacks due to COVID-19 across the research space, it's likely that the failure of new unicorns in the tech space and excess capital across the board will result in continued investment in various gene therapy methods and rare disease single gene targets. However, the recent deaths highlight the catastrophic consequences and massive challenges that face development in these therapies, even when robust preclinical data exist. The study in question was sponsored by Audentes Therapeutics, which is currently owned by the Japanese multinational company Astellas Pharma, whose shares have remained relatively stable around $16.50. On June 30th, the FDA released new regulations for all COVID-19 related vaccines. This report does not represent the final determination of what the FDA will want for approval. However, a critical guideline from the report was the requirement for 50% efficacy for any approved vaccine. In this context, efficacy may include decreased symptoms, severity, and duration, as well as the more obvious prevention. Some experts consider the 50% too high, while others suggest it may be too low. For comparison, the annual flu vaccine is usually 40 to 60% effective. In general, the 50% guideline likely indicates that the FDA is trying to balance the need for a vaccine soon with the desire for it to be reasonably effective. Keep in mind that a recent AP poll suggests that only 50% of Americans will get a vaccine if it's available, meaning the public health challenges may outpace the scientific ones. The current frontrunner in the COVID vaccine race is Moderna, whose stock went from around $20 a share in February to $58.57 at closing on Thursday. The cascade of novel CRISPR-related technologies continues this week. CRISPRs function as part of bacteria's immune system by creating targeted DNA cuts. Like all things with evolution, a good defense often leads to new methods of offense. A paper out July 3rd in Science by Alexander Misk and colleagues demonstrates a novel method of viral offense against Cas13. They discovered a Listeriophage virus that produces a protein ACR-V01, which inactivates the function of the CRISPR protein Cas13. For the virus, this means it can survive the defense system and successfully replicate. For scientists, co-opting proteins like ACR-V01 may provide a means to activate and then deactivate a CRISPR system. This can theoretically lead to more temporally precise gene editing methodologies down the road. Imagine being able to precisely edit a specific DNA sequence and then turn off that machinery forever so you don't have continuous editing. there are extensive possibilities given enough biotechnical refinement. Thanks for joining me on the Niche Podcast. Our goal is to keep you informed so you can engage your colleagues. Join us weekly for a 5-minute summary of top news in the biotech, clinical trials, and applied science industries. You can find us on your favorite podcast app. Like, comment, subscribe, and most of all, share with your friends in the industry. Once again, I'm Dr. Noah Goodson. I'll see you next week.